very happy that we're back in action live and in person. I think, so far all the seminars that we have lined up are going to be in person, so we're real excited about that. So as we've done for the last several years, we have our own faculty present their own research, giving you a very short synopsis of what they're doing, 10 minutes. We have a bet with Professor
All right, good morning. Welcome, and I'm happy.
Class, right?
It is, yeah, I tried them to be here. So I'm happy to talk a little bit about our research. I'm going to try to go quickly, very overview level, high level of what we do in the lab, just to kind of give you a flavor for it, and happy to discuss any of these things in more detail if you like. But our lab really focuses on cardiovascular disease. We're looking at ways to both diagnose and prevent or treat cardiovascular diseases. So we'll start out with some of our story about treatment. And we like cardiovascular disease because we're really interested in tissue mechanics in an area called mechanobiology. So how physical forces cause tissue to move, and how those, when that tissue moves, how the cells feel those forces and then transduce that into some biological response. So both arteries and aortic valves, two things that we work on a lot, they move 30 million times a year. Stretch and relax as the heart beats and relaxes. And we focus a lot on a specific pathophysiology, something that happens in these diseased tissues, which is cardiovascular calcification. And that's the formation of bone-like mineral that forms in cardiovascular tissues. So you can imagine that if you have these soft tissues that are supposed to be moving all the time, if they start turning into bone, they don't move so, right? And it turns out that the amount of calcification that you have in these tissues is the best predictor of whether or not you're going to have an acute cardiovascular. It's the best predictor of your long-term outcomes. This is especially the case in people with chronic kidney disease. So chronic kidney disease leads to widespread cardiovascular calcification in many patients. And the more calcification you have, and this is looking at aortic medial and intimal calcification, but basically the story is the more calcification you have, the less great your outcomes are. So this is survival rates, right? So people with no detectable calcification in end-stage chronic kidney disease, almost all of them survive out to whatever 100 times divided by 12. It's almost 10 years, right? Whereas if you have intimal calcification in aorta, only about 30 to 40% of those people are still alive within 10 years. Okay, so it's a big problem, and it's really just due to disruption of the mechanics in these tissues. So this patient population in particular is in need of anti-calcification therapies. We think this is going to be a good place to start trying to identify therapies for this population that we could then extend to a more general population that also gets calcification. So when we zoom in on calcification, so this is actually from a human atherosclerotic plaque, and we zoom in on the calcification. This is using SEM. The color here represents the density of the material. So the more orange the colors, the calcification. The green colors are other organic materials like collagen, okay? And you can see that the calcification is not very smooth. It's not homogeneous in the way that we think about maybe bone. It's actually made of these smaller vesicular structures, on the order of hundreds of nanometers. And it turns out that what happens in these tissues is cells, when they are exposed to pathological conditions, they become bone-like, and they release these little vesicles, these 100 nanometer size vesicles. These very, very small vesicles that are really good at binding calcium and generating free phosphates, the two components that make mineral. And they get those things together in a really small space, so they start that nucleation process, that mineral formation process. So this is some work we did with a collaborator of mine in physics a couple years ago. This is all computational simulations to understand how this works. But then one thing that we've gone on to do is to look at the mechanisms by which those vesicles form in the first place. These cells release these vesicles. And so we go back and say, where in the cell do these vesicles come from? And can we figure out how we could then stop that process? The reason we think we want to stop this vesicle process is a lot of things can cause this to occur. Chronic kidney disease, high blood phosphates, hyperlipidemia, hypertension, And treating all those things can be very difficult. Sometimes it's multifactorial. And so we're wondering if we can target this specific process where a lot of these things converge, we might have a more universal therapy for vascular calcification. Okay? And there's years that have gone into identifying different components of this pathway, but one thing that we've keyed in on recently is targeting a receptor called EGFR, okay? Epidermal growth factor. And one reason we've targeted that, this is from a review paper a couple of years ago, this is a very accessible target. So if you're ever in the process of developing new drugs, one thing you always have to think about is, could I actually make a drug that can reach this target? And in fact, this receptor is targeted clinically now in chemotherapeutics. A lot of cancer patients take EGFR inhibitors. And so we know that this could potentially be a good target for us. And we have reason to believe that it would alter the formation of these calcifying vesicles. So we do a lot of study with mice, and we actually give these mice chronic kidney disease by feeding them a diet of high adenine. And this adenine basically destroys their kidneys. So on the right here, these are cross sections of, or histological sections of kidneys. This is how it should look. You see all these vacuoles in here of both vehicle treated groups. This is a control group, these mice that just don't receive any drug, and mice that receive an EGFR inhibitor. Okay, so we're trying to give these mice an EGFR inhibitor to see if we can stop the calcification process. And what we show is that it doesn't stop the dysfunction in their kidneys. Okay, so they still get this chronic kidney disease. So these mice receive a high adenine diet for a long period of time, and they receive a high phosphate diet, which actually causes that calcification. And then we inject them with a dye that binds minerals. We can track everywhere in their arterial bed that they have calcification. We can track it and quantify it. And so these are resected mouse aorta. So this is a mouse that didn't get chronic kidney disease. You can barely see it here. The dye just, the more intense the dye the signal we get from the dye is the heat map, so it's shown in more yellow. So this mouse has very little calcification in its aorta. This is the chronic kidney disease mouse, and you can see how much more intense the calcification is in the aorta. And when we treat with that EGFR inhibitor, and this is quantified here, we can see this EGFR inhibitor significantly reduces calcification in the aorta of these mice. And we can look at cross sections as well. This is using a different stain. The red here is calcification. So you can see all this calcification in the arterial wall of the CKD mice, which is completely absent in this section from the EGFR treated mouse. So this leads us to believe that this could be a good target for vascular calcification. One thing that we wanted to check was, if you're talking about inhibiting mineral in the vascular system, what other tissues mineralize? The bone, right? And you don't want to inhibit mineral formation in the bone. And this is CT scans of bones from those same mice. And we've quantified this as well. And in fact, CKD, the chronic kidney disease, causes decreased mineralization. It seems like the inhibitor actually reverses that even a bit. So if anything, we have a positive effect on the bones of these mice. So it seems to be specific to the vascular system. Okay? So that's really exciting news. And most of this work was done by, I can't use my pointer, by Amarala, who's a senior grad student at the lab, who's graduating soon, with the help of Hui, who's a postdoc in the lab. And I put Anna here as well. So Hui and Anna are working on a different molecule, small molecule, to do something somewhat similar. So we're looking at different therapeutic targets for this disease in the lab. I mentioned to you earlier, I just want to throw this in here, that one thing that we look at is mechanosensitivity, how mechanics affect cells in these tissues. And it turns out caveolin-1, which is what EGFR has interacted with, is mechanosensitive. We also have some data showing that the more these cells are stretched, the more they release the vesicle. So moving forward, we're going to try to understand how mechanics play a role in this as well. So, and then using our targets to prevent things like hypertension-induced calcification. So that's one half of the lab. The other half of the lab is really focusing on the diagnostic aspect. And it turns out that these diseases are severely underdiagnosed, the ones that we really care about, especially in underserved populations. People who don't go to the doctor every single year for checkups, all right? And so what we're trying to do is we're trying to develop point of care technologies, low cost point of care technologies that can be used to diagnose these patients. So this is one effort in the lab where we're looking at circulating factors that are in the blood that are known to be associated with these diseases, and developing microfluidic platforms where we can say put a drop of patient's blood in this platform and then look for these specific markers to tell us how at risk is a patient for having cardiovascular disease, for having cardiovascular calcification and things like that. So this is ongoing efforts led by Lin Tong and the PathSub ERC. We're also interested in aortic stenosis and calcification of the aortic valve. And we're looking at how this remodeling affects the mechanics of these valves. So these valves, again, just like the arteries, you get calcification, they don't move as well, okay? So we do a lot of biomechanical characterization of these valve leaflets. But what we're trying to do on the diagnostic side is then use the sounds that are heard through a doctor's stethoscope. That sound is created by the leaflets of these valves, these tissues, banging against each other when they close. And we're looking to say, can we pick up signatures in that sound that tells us that these leaflets have remodeled, have calcified, so that we have a subjective, non-invasive way of identifying patients with aortic stenosis at the point of care. And so we're using those same mouse models. We have a stethoscope, we put a tiny little bumper on, and we have these little mouse that have chronic kidney disease. We put them on their back, and we use a stethoscope and measure the, we record their heart sounds. And then we're developing, with the help of Dr. Danziger and some others, machine learning analyses to understand how signatures in those sounds correlate to the disease present. So this is our group. It's a lot of fun. We've been fortunate to be funded by the Florida Heart Research Foundation, the National Science Foundation, American Heart Association, and NIH as well. So happy to take any questions.
How specific is the EGFR to Gavioli?
Yeah, so EGFR is a known cadmium-1 interactor. How specific is it to that potential? Like, so would there be off-target effects? Perhaps, right? And we don't know yet. We know there's none in the bone, and we know that this isn't affecting other types of vesicles released from the cells. We've looked at that too. And we know it's well tolerated, at least in cancer therapeutics. So I think there's a lot of reason to believe that it could potentially be developed into mechanism, but maybe some targeted drug delivery modalities would be pretty good too. Yeah.
So what kinds of cells would be the target for?
Yeah, we're looking mostly at vascular smooth muscle cells. So the vascular smooth muscle cells are the ones that seem to be releasing these vesicles that cause that mineral form. They actually start looking a lot like osteoblasts in these diseases for reasons that we don't quite understand.
Like mesenchymal cells?
Mesenchymal cells, yeah, exactly.
Those would be the ones like undifferentiated and differentiate into bone cells?
That's right, that's
so
right.
That would be like mesenchymal cells?
Exactly.
Like fibroblasts?
Exactly. So yeah, so it could be some resident fibroblasts, it could be, you know, like I said, the arterial bed vascular muscle cells, which are also a mesenchymal type cell. Yeah.
So that PK?
Not in this particular study. We have a different study that does show that most of these cells that come into the vascular bed and calcify are from actually the from adventitial mesenchymal origin. They acquire a muscle cell phenotype before they go and calcify. So, but we haven't gotten there with this particular study yet. But since they are the major contributor to that calcification, they're likely to target. That's pretty in the weeds.
Yeah, I think you may have just answered this, but what is the non-pathological purpose of vascular?
Yeah, so that's a good question. And honestly, we don't know. There don't seem to be exosomes. They may just be a specialized vesicle that's released just for this purpose. And we think the reason they're released and the reason this actually happens is that in some, so your body, like if you have a traumatic glass injury, the wounds there will actually calcify often. And we think that it's a wound response mechanism. These cells are trying to just kind of wall off a certain area of the body that's been wounded. But that doesn't work so well in these tissues that are supposed to be so dynamic. So it's just supposed to be a generic, like, you know, oh crap, this, you know, things are going wrong. Let's try to stiffen the whole extracellular matrix up.
Okay, so Wei-Chiang Lin from the BME department, and I called my lab Biomedical Engineering Creative Lab because initially my research focus is on biophotonics. Over the past probably 5-10 years, it has diverged to different areas. So and I thought, okay, since I'm in control of the name of the lab, so I could change it to whichever one. Okay, so the research activities in my lab in general fall into 3 sub areas within biomedical engineering. Those 3 areas are probably less exposed for most of the students: biophotonics, imaging, and instrumentation. So over the past few years, because of the addition of the new projects, so the map expanded a little bit to toward the biomechanics, biomaterials, and the computer science. And everybody's using artificial intelligence, data mining, etc. But I'm also interested in virtual reality and augmented reality. They are relevant to some of the research projects. Okay, so one major concentration, research concentration in my lab is non-invasive tissue diagnosis. So we develop optical, mechanical tools or technologies that can measure the intrinsic properties of biological tissue, and use that information we can differentiate disease and injured tissue from normal ones. So using those technologies, we have developed surgical guidance systems, and then we also develop continuous physiological signal sensing tools, and for various applications. And I think probably over the last 4 years, another new concentration is formed within my lab, and that's orthotic prosthetic technology development. So we develop sensors and the devices to capture motions and the movements of the human body to understand how we can better design prosthetic orthotic devices. Okay, with that said, there are just so many projects for me to highlight. So that's the difference between research activities of junior faculty and the senior faculty members. So senior faculty members can explore different areas, with no repercussion. So I also just highlight a couple research projects, which I think really, exemplify the research activities in my lab. And so the first one is all optical quantitative ultrasound imaging system, and then the second one is implantable CO2 sensors. Before we talk about those 2 projects, I just want to give a shout out to my PhD students. They work hard, so I look good, and so I can give this nice presentation. And I'm pretty sure that's true for all the professors. Okay, so the first project I want to highlight is the all optical quantitative ultrasound imaging system. So there's always a reason you want to develop a new technology for tissue diagnostic modality. So for a very long time, we have been working on this surgical guidance system for brain tumor surgery, and it's optical based. Has great features, resolution, sensitivity, great, but there's one Achilles heel, and that's the penetration depth. That's unfortunately true for all optical devices. The penetration depth is limited because light doesn't penetrate deep into the tissue. So information millimeter beyond the resection front is not available. So So even though your device works very well for certain conditions, we feel that particular limitation has to be addressed in order to make this system practical for neurosurgeons to actually incorporate during the surgery. So long story short, we start looking into a new modality we can integrate easily into these optical surgical guidance system at the same time that provides penetration depth or the actual resolution. And that's all optical ultrasound. So some of you probably have taken Dr. Makura and my imaging courses, so you know what ultrasound is, right? So to create all optical quantitative ultrasound, you have to have 3 elements. You have to have an optical transmitter, I call it OUT, and then you have to have an optical ultrasound detector, OUD, and then you also perform quantitative analysis to echoes. So typically in ultrasound imaging, echoes are analyzed according to the intensity. So we call that echogenicity. And we feel like we can do more than that. Okay. So I have these slides to explain a little bit in details. We use photoacoustic effects to generate ultrasound waves. And so you shine the laser lights into a short medium because energy conversion from photonic energy to acoustic energy under 2 confinements, thermal confinement and acoustic confinement. And to detect ultrasound, the few mechanisms available. So the first mechanism we explored is refractometry. So basically, when pressure wave propagates through a medium, pressure induces a local change in the refractive index. And the refractive index changes the way light propagates. Okay. So my PhD student did an animation, but I couldn't incorporate into this slide. But nevertheless, the mechanism is simple, but it's extremely sensitive. Sensitive than the conventional, say, ultrasound transducers like PZT transducers. The last one, we didn't create this. Somebody started working on this a long time ago. So echoes, if you look at the RF signal, echoes actually is oscillations, oscillation at the ultrasound frequency and in the particular frequency. And typical ultrasound imaging systems convert this signal to envelope, and the envelopes then converts to the intensity information. However, when you convert this echoes into an envelope, you lose the frequency characteristics of the echo. So our imaging device will preserve that information, use that to perform diagnosis. Why? Because the reflectivity of the biomaterials or biological tissue and is reflectivity of ultrasound and is frequency dependent and is tissue composition dependent. We feel like there's another window of opportunity we can utilize to perform tissue diagnosis. Okay, so advantages, and it's tunable, has a tunable central frequency, and which means that you can control the resolution, and you can also control the depth of the imaging, on the fly using the same transducer. Also has a very broad frequency bandwidth, so that means that it's really suitable for ultrasound. It's feasible to miniaturize such a sensor and make it easy to incorporate in intraoperative environment. It doesn't use high voltage, which typically we see in the conventional ultrasound imaging systems. So electrical safety was a big no for it. The last one, which is something we try to demonstrate, Okay, so it's almost like a sci-fi movie in that we're thinking about the feasibility to perform ultrasound imaging without being in contact with the patient. So that means that if we can realize that, that mechanism, I'll be able to scan your body from a distance. Okay, but we're not going to talk about that, at least in this presentation. So this is what my previous PhD student, Muhammad, was able to accomplish. So we built this all-optical quantitative ultrasound transducer, and we use that to scan phantoms with a different composition, i.e. different scatters. So we were able to demonstrate, first of all, we can create BMO image with the ALL-OPTICAL system, and we also have the capability to perform quantitative analysis to the echoes and hence differentiate phantoms with different scatter size. Okay, so the second project I want to highlight is Hutcheson's project. I think Hutcheson probably is in the audience, not here, but in the virtual audience. Okay, so implantable CO2 sensor. I'm going to speed up a little bit because I think I'm running out of time. Okay, so this is a diagram I copied from Dr. Brown's lecture, and this shows the regulational system in the normal human subject. Right, it's a complex system. It's a closed loop. When you have injury or disease, components or this particular closed loop system will be impaired. Some components will be weakened, or the entire loop will be destroyed. Okay, so specifically spinal cord injury. So for patients with impaired ventilation regulation, so doctors usually subscribe, technology such as mechanical ventilators or diaphragm placing to help them breathe. Okay, but introduction of those two components into the picture does not reestablish the closed loop nature of the ventilation regulation. So there are consequences, bad consequences, and so solution we propose, we envision, is to build a subcutaneous capnography, and so provide feedback. And so what is a subcutaneous capnography? So the cartoon on the right-hand side illustrates the device that we built to measure interstitial CO2 level. So it has two components. One is an implantable CO2 sensor, and the other one is an optical reader. Implantable CO2 sensor is placed in the subcutaneous space and measure the PCO2 in the interstitial fluid. And then the response of the CO2 sensor is optically read by the optical reader on the tissue surface. So to build a system like this, we have to make sure the biocompatibility the sensor is sensitive to CO2 only, and the CO2 sensitivity should be in the range of the physiological range. So that's why we have specs. And to make the system work, this sensor has to have such a quality in order to make this subcutaneous capnography work. So how do we design a implantable CO2 sensor? We didn't create this sensor from scratch. We actually drew inspiration from Sorenhouse CO2 sensor design and create this implantable version. And so we have a sensing medium, and which is sensitive to CO2, and we wrap it with a membrane, and the membrane has a selectivity to CO2 gas molecule. So this particular picture illustrates the three mechanisms utilized by the implantable CO2 sensor. And the diffusion, which allows describes how CO2 molecules diffuse from interstitial fluids into the sensor or sensing medium. And inside the sensing medium, we have a chain of the chemical reactions. And then lastly, we have light sensor interaction, which is absorption or fluorescence emission, and that gives us the optical signal, hence the sensor response. So knowing those mechanisms allow us to build a model to simulate the response of the CO2 sensor to the PCO2 in the environment where the sensor is placed. So Hutcheson built this model, and so we were using that model to predict the sensitivity of the sensor with the different configurations. And we also spent quite a bit of time to understand the response time of the sensor. Serenhouse CO2 sensor tends to be slow. Slow, I mean minutes or hours. And but that's not good for physiological or biomedical applications. We need to speed that up. So we designed and carry out an factorial experiment to understand how sensor parameters, thickness of the membrane, concentration of sensor medium, and etc. interact with each other, how they impact the response time. And if you're going to take the data evaluation to understand what factorial experiment design is. Okay. Too much data to show, but I'll highlight just one. And actually, this is a preview of what Hutcheson's going to present at the end of this conference. So one of the interesting findings is that the selective membrane, which is PDMS, and the diffusivity can be tuned by the composition of the PDMS. So we were able to demonstrate that by changing the curing ratio, i.e. the ratio between curing agent and the base polymer, we can speed up the sensor. Okay, and so far, we were able to achieve sub-minute response time for our CO2 sensor, but we are pushing it toward probably seconds, a few seconds of response time, which we think will be suitable for biomedical application. And there are a lot of results I cannot introduce in today's presentation. But if you're interested, I think definitely contact Hutcheson, and he'll be happy to give you a comprehensive explanation about the entire project. Okay, so I think I have run out of my time. And but nevertheless, I hope I have select the right projects to entice your interest in research activities in my lab. And again, I'm always looking for inspired students to work with. And if you're interested in anything mentioned in today's presentation, feel free to contact. We'll have to discuss. Okay, so that's the end of my presentation. And questions? Yes.
What's the chemical reaction that you guys CO2 sensor? What do you guys target?
So the sensor medium basically is a water-based solution. So basically, when CO2 diffuses into aqueous environments and it becomes carbonic acid and changes pH. Then we have optical pH sensor embedded, and that basically reports pH, hence indirectly report the CO2 concentration in the sensing. So that's a brief.
What's the chemical in the sensing material that produces the light change due to pH?
So there are two pH sensors we're using. HPTS, I don't remember the full name, and the other one is Cipher 5, which is in the near infrared wavelength regime, which will be the final sensor.
So they change their absorbance based on pH?
They change, yeah. So for HPTS, and HPTS changes its absorption property according to pH.
Is it the amount of absorption, or is it the peak wavelength of absorption that shifts? Which is
Both. So it also has fluorescence property, which we have not tapped into, and because absorption leads to the change in the fluorescence property. Cipher 5, if I remember correctly, is absorption based, and it does have fluorescence property, but it's not as sensitive to the pH variation as absorption.
Is it reversible?
Huh? Reversible, yeah. Any questions about yes. Yeah.
How small could the device get?
I think
How small is it?
possible. So it's a cube right now, and it's about probably 2 centimeters each dimension. So that's the dimension.
What do you think you can get it to?
We think we can eventually create a version, probably small enough that you can fit to the tip of a surgical tool. But that's not.
Surgical tools can get pretty small, because now
That's why I'm trying to be vague. I really don't know how small, but that's not the direction we want to push. I think the direction we want to push is the non-contact ultrasound image. I'm going to paint the picture, and I don't know how realistic that is. So surgeons, during the operation, they're working under the microscope. Okay, so they want to see what's beyond the resection surface using our ultrasound image modality. So they will cover the resection front with some type of material. And then we'll send the laser light through the microscope and to excite the membrane, which is on the tissue surface, that generates ultrasound. And then the same membrane detects ultrasound optically, and that signal will be picked up through the microscope. So basically, that's one way to use all optical quantitative ultrasound. We envision. How long would it take to realize such a vision? I don't know. Depends on how many bright students I have.
Now it's funny.
I know.
Good morning. So I am the third speaker for today, and my topic is on heterogeneous package integration. It can be used for a lot of things, but what we use it for is health monitoring. And I see some. So what is package integration? Package integration is putting all these system components together to perform the system function. It is basically system integration, so-called hardware integration. It's very important for biomedical devices. Hardware integration drives advances in implants. People come up with new therapeutic techniques, new devices, they come up with new models. At the end of the day, where it goes to reality is when you put everything into a system hardware that works under the clinical constraints, manufacturing constraints, and the performance metrics. So it's all about hardware integration that we deal with. It involves putting devices together, providing a protection around them, delivering the signal and power to the target locations through the feed-throughs and connectors and wires, cables, electrodes. And you can see the systematic evolution. I didn't put any arrow because everything has its own merit, and they will have their own sustained presence in the biomedical industry. But you can see that the traditional ones are more like pacemakers, been there for decades. Recently, more implants that are used for deep brain stimulation, retinal prosthesis, other kind of neurostimulation, where you're getting more and more higher densities of interconnects between the devices to the electrodes to the target location, the electrode densities, and that requires more functionality, efficient power delivery, ability to provide more reliability in a smaller scale with higher densities. Eventually, what we like to do is a 3D version of this, where we can integrate multiple devices. Instead of using bulk titanium cans, can we use very thin metallic ribbons or ceramic ribbons that are now coming in the market? Can we provide feed-throughs that can be at a less than 50 micron scale instead of several hundred microns, so that you can target more locations for your sensing or therapy. So, and can we assemble these things with innovative bonding techniques? So those are the things we are interested in. And not only for implants, we are also interested in this for wearable health monitoring systems, where we see more industry sponsorship for us today. And actually, most of the research I do is sponsored through industry funding and partially through federal. So in the wearable systems where there is a lot of drive, wearable health monitoring systems, you can see that today's systems utilize more traditional system integration techniques, where devices are initially packaged, then you put them on a large PCB, and then you encapsulate them and then assemble them on these flex electrodes. And they're getting to be smaller because of these package integrations more than anything else, because the devices are getting efficient and the co-packaging or integration is getting more efficient. And what we like to do is something towards the end where we can integrate these devices within the flex electrodes, providing the power, data, reliability, everything. So what does it entail? What do we mean by, it involves a lot of things. It involves delivering power. So we are interested in power telemetry. How to deliver power, it involves data, how to get the data out from these systems, packaging of these photonic devices so that we can get more modalities, multimodal sensing, so that you can use efficient algorithms to accurately stratify the diseases using machine learning technique like what Dr. Hutcheson just mentioned. Very high density feed through so that you can feed a lot of electrodes at the same time. And advances in the electrodes themselves so that we can provide lower impedance and more stability, and integrating all these functions. So it's a pretty ambitious program that I try to lay out here with a lot of technology elements and how to bring them together. And it also involves some aspects of simplifying the system topologies. Even if you have all these technologies, if you want to put 100 components in a very tiny flex package, it's gets to be expensive. So can you simplify the topology so that you can minimize your bill of materials, and also enhance with advanced material technologies? That's what we focus on, how to improve materials to integrate them better so that we can deliver these functions. So what are we doing specifically? My original background has been in the last three items, how to provide these high density feed throughs, encapsulation and bonding, and low temperature assembly or welding of these packaging materials. But I got more and more attracted towards the first three items. So I am trying to go into more RF power telemetry and advancing the power densities and high density device integration to provide this either delivering power or retrieving data using new ways of package integration technologies. So I will still get back to this eventually, but most of the things we do are in the top three: how to enhance components to deliver more power, retrieve data with lower power in a smaller form factor, make it more scalable, lower cost. And so again, I show basically traditional packages here that are in bulky cases with very thick circuit boards, and more emerging technologies where you are having them on a single chip device, all the system functions with some nanoscale protection coatings. But these actually, the real solutions lie somewhere in the middle. These are, they cannot provide the whole system functions. These are too bulky and more yesterday's technologies. So what we are trying to do is some combination of hybrid techniques that are easy to realize. The other aspect of it is about smart and defect-free manufacturing. Can we create a manufacturing process where we can monitor the defects? Can we heal the defects in situ while it's being manufactured? And can we create some mechanisms that can enable self-healing of the defects so that we can create a defect-free, advanced, highly miniaturized flex packages that can be used again for the same wearable health monitoring systems, implantable health monitoring systems, neurostimulation or other kind of therapeutic techniques. So the applications range quite a bit. I will talk about some of the applications because only in the context of the application the package is relevant. The packaging or the system or integration is only relevant in the context of the specific clinical need. So I will talk about that in the next few slides, but what I like to highlight is using certain innovative biomedical knowledge, we can actually advance manufacturing science. Just like the way we advance manufacturing science to heal diseases or to advance biomedical engineering, we also do it the other way around because of my background in both manufacturing, electronics, and biomedical engineering. So we also try to create some nice defect-free manufacturing techniques. The first thing is about embedding devices in a very thin flex package and providing these connections. So we talk about the devices that are buried inside the package, so that they are flat with the surface. They are embedded so that you can put multiple layers of them together, you can stack the devices, and hence you can create this wearable health monitoring systems. So these are some examples of what we recently did. And the first application that is driven by Yahya, who is going to hopefully do his proposal in a few months, is about wearable health monitoring system like wireless ECG, even though he also has interest in other recording, for example, from the brain, but we are targeting wireless ECG, where we make this very thin patch and stick it to the electrodes. And this patch basically has a lot of new nice attributes. It doesn't need any capacity, it doesn't need any power storage, it doesn't need any active devices that consume power. It's purely passive, and hence you can use it for very scalable and cheap neural recording techniques. And we demonstrated this using advanced packaging. This technique by itself is not developed by us. It's actually from Professor Wallace, and it's also used by Carolina and Dr. Riera. But what we do here is advance the packaging of these devices by embedding flex integration, miniaturized antennas, so that we can make these flex packages more scalable, better performance, and more efficient in their in the power that they require for continuous monitoring. For example, these devices always need an interrogator nearby to continuously send RF power and retrieve the RF power. So how to minimize that power is a big need. The second thing is, you still need a lot of devices that consume power. So how do you deliver power? The usual way is inductive links, coils. You want magnetic coils, and you couple them with near field inductive links, magnetic field coupling, and you deliver power. But you run into problems with delivering a lot of power in very tiny millimeter scale devices, like for example what we are talking about. So we need to invoke new kinds of power delivery. How do you deliver power? And we are basically moving away from these large coils to very tiny chiplets that we can deliver power, milliwatts of power, very efficiently at low frequencies, so that the body does not absorb. You have more room to send power to the body at lower frequencies. And unfortunately, you can make, using this multi-physics phenomenon, like the so-called multiferroic resonators, piezomagnetostrictive resonators, we can deliver power in millimeter scale, and you can perhaps deliver a few milliwatts. And that can be now used for neurostimulation. It can be used for powering photonic devices like what Dr. Wei-Chiang and we work on. And so it involves bringing multiferroic materials like a piezomagnetostrictive stack, a piezoelectric material in combination with a magnetostrictive material, so that you can work them in the resonance mode. You can make very tiny resonators and deliver power. And we recently got the first demonstration of this resonator working. And now we believe we can deliver this kind of power. It can also be used for other applications. So we are also working on using this for even data telemetry and other applications. And this is driven by Yahya and also the new student Viru Jaswal, who joined us about a year back with a lot of background in biomedical and microbiology. So he's trying to apply these kind of techniques for these devices. The next part of it is basically you need to enhance the piezoelectric materials, but you need to make these magnetic films compliant with the piezoelectric materials, so that you can bring these multi-physics based biomagnetic sensors and also deliver power with these magnetic films. So we are also using these magnetic films for efficient power delivery. And the other option, the other application is to create innovative magnetic therapies that I'll talk about. So all of them requires very advanced magnetic films that we are working on. And again, there are 3 students that work on this. Carlos, he recently graduated, and Viru also works in this because it's coupled with this piezomagnetostrictive, and Juan Gomez has been actively working in this project. So this is how we create these multi-ferroic stacks so that we can make innovative wireless power transfer and data telemetry, and also new kind of magnetic therapies that I will briefly discuss in a minute. So once we have this piezomagnetostrictive materials, we can come up with a new ways of health monitoring. For example, you always know about wireless ECG, electrocardiogram. Then we do a wireless SCG, seismocardiogram, where we can understand the cardiac mechanical events in a more detailed way. For example, you can study the onset of diseases with this seismocardiogram, and that is what we are now fabricating with our PVDF magnetostrictive films by micromachining this PVDF layers and then putting very thin, flexible, compliant layers, films, and perhaps we can now monitor them wirelessly with 0 power using the same topology that I talked about that we use for wireless ECG. We now want to make the first Band-Aid-like, very miniaturized, flexible wireless SCG. And I think Dr. Hutcheson has a lot of interest in this, so we also like to collaborate. So that is one project that we are very excited about. And you can also use this piezomagnetostrictives for harvesting power from the body. Though there is a lot of ambiguity about how much power you can generate. Is it really useful? Can't you just use a battery? But if you actually use it efficiently, and if the devices are designed well for low power, you can perhaps use them also for efficient power harvesting from the body. And the other aspect of it is we are also enhancing the electrodes to improve the impedance compatibility, lower impedance electrodes. And that's a project that the undergrad student who just graduated recently, he worked on. They are trying to develop some new low impedance electrodes that meet all the other criteria like mechanical compliance, mechanical robustness. And one aspect of it is porous tantalum wires that we can surface modify to create these low impedance electrodes that can meet these criteria, where you can get lower impedance and again higher charge injection capability if you're using it for neurostimulation. The other aspect of it is about connectors. Once you have this package and you need to deliver the signal to a bunch of locations using electrode arrays, you need connectors. And these connectors have to be rematable in a surgical environment. That's a big deal. Right? How to make these connectors that can be rematable. And a lot of advances in this. Traditionally, you have more these bulky connectors that went into these spring connectors. And Dr. Jung from FIU and Satya, and they invented a new set of connector technologies, the multi-channel connectors. And we are using more area array connectors using our Z-elastomer feed-throughs that we developed in our other research. So we can make these connectors perhaps more higher density and again meeting all the requirements, the hermeticity and long-term reliability and compatibility with surgical constraints. And the last aspect, again, I briefly want to highlight is what we are doing with Professor Wei-Chiang Lin about the biophotonic packaging. Sept has been working on it for a year. Since we have these high density flux packages, we can deliver power, hopefully retrieve data, and can we use them for biophotonics? And that's one project that we also work with the senior design team. So some of these results actually are being published in a conference from the senior design project. How to use these advanced flex packaging techniques for biophotonic applications. The last thing is, can we use these innovative magnetics for therapeutics like magnetic therapies? And one thing that I'm interested in is this magnetic decalcification. Can we suppress the calcification in the heart valve or the cardiac tissues using magnetic fields? And traditionally, there is use of very large bulky magnetic field generators, and that's not what we're going to do. We are going to now integrate these magnetic films into very tiny flex cables so that you can create these magnetic fields in a more efficient way. So as I said, most of the research is funded by our industry partners. So we work with industry, which is very difficult. We have to be, attentive to them almost every day. And we have so many bosses, right? Every industry comes with 2 bosses. Every project has like 2 supervisors that work with us, and very demanding. And we have 5 of them. So obviously it's very, very time consuming to work with these industry projects, and they don't pay a lot of money, unlike NIH and DARPA. So it's a lot of work for little money, but it makes us focus on the real problems. We do have NSF funding from EAGER, ICOPS, IUCRC. That's a new industry partnership program coming up here. So we work at least in close partnership with commercialization. And this is the team. I think we have quite a few students who did work, and some of them graduated. One of them is Carlos, who just graduated, and he had 3 industry offers, and he had tough time choosing which one to select. So it's a good problem to have. And that's the good thing about working with companies. You're always in touch with your industry partners, so easy to get jobs later. So we have some advantage. So anyway, this is the kind of research we do: advancing system hardware, package integration to solve wireless neural recording, biophotonic package integration, magnetic therapies. Everything requires power. Everything needs to retrieve data. And whenever you have this power and data, and a very reliable, very challenging environment, you need a lot of system integration advances. That's where packaging comes in, and that's where we play a very broad and critical role. So if you're interested in any of these technologies, power, data, photonic packaging, flex packaging, electrode integration, please talk to us, and we'll be happy to work with you. Any questions? I think I took 20 minutes to.
So for a student that wants to be involved, how much do they need to know about circuits and electronics? And
Not really, not much. Actually, we work with devices, and we interconnect and power them. So the basic knowledge of circuits is adequate. You don't need a lot of details about any IC design. You need to know basic package layout, and that can be very easily trained, very easily taught. So most students come with very little knowledge, and they learn and they pick up. So you really don't need a lot of circuit knowledge to advance the hardware integration technologies.
so the circuits course that our students take.
That's adequate.
Should be
They should know basic about power and data layout.
So for those wireless device, self-powered wireless devices, what are the external sources we have to use to power those, those devices? Do we have to have a RF source, or?
So there are 2 things. One is the telemetry. If it's a piezomagnetostrictive, you need to generate a few kilohertz of magnetic fields. That's power telemetry, and that can hopefully generate milliwatts in a millimeter cube dimension. That's what we are targeting. For harvesting, it's from internal energy sources, like the cardiac mechanical waves are basically harvested to generate. The piezomagnetic strictive is so compliant. The way we micro machine this PVDF films, very thin PVDF films on the skin, they are supposedly so compliant that you can pick up a few volts just with a Pascal of mechanical wave. That's the target that we are looking at, 10 volts per Pascal. And the body generates that kind of mechanical pulses, so you can harvest power from those mechanical pulses. Whether you can get to several microwatts, 100 microwatts, that in a reasonable area, that is what we are looking at. Unless you get about 10 microwatts to 100 microwatts, you can't really do much with it. So that's, at least you can do some wireless neural recording with that. So that is what is our industry driving us to one partner at least.
Other questions? All right, then I guess we'll wrap it up for today.
Thank you.